We'll begin our look into the nervous system, looking at the basics of the nervous system itself. And so when it comes to communication in the body, the nervous system is our primary way. It's the fastest way to get a signal from one place to the other. The only thing is that in terms of the nervous system, we have to keep sending that signal if we want essentially a long-term solution to that information that needs to be sent. And so we can divide up the nervous system, much like we've done with our other systems, in a number of different ways. And so one of the first ways to divide it up is where we find things. And so central nervous system versus peripheral nervous system. The central nervous system contains our brain and spinal cord. And so it has those two basic components within it. Functionally, this is where essentially all the processing occurs. This is where we're taking all the information in and sorting it out and determining what we need to do with it. And so this is essentially where we have input of sensory information and output of motor information after we have dealt with the information that's there. So this is all of our intellect, this is all of our memories, this is all of our learning processes, anything and everything that goes on in terms of the brain and processing of information is happening within the central nervous system itself. From there, we have the peripheral nervous system, which is everything outside of the brain or spinal cord. So cranial nerves, spinal nerves are the way that we're going to get that information to and from the central nervous system itself. And this is going to carry both a sensory side and a motor side of information. So we're going to have an afferent and an efferent pathway within the peripheral nervous system. Functionally, we can divide things up into sensory versus motor. And so essentially in the peripheral nervous system, we have neurons that are going to be carrying this information back and forth to and from the central nervous system. And then once we get to the central nervous system, we'll kind of differentiate that information and send it to the proper places within the central nervous system as well. So sensory information we call the afferent information. And so this is information that's coming back in towards the central nervous system. It can be divided up into either conscious somatic information, so we have the somatic sensory information, or it's primarily unconscious visceral information, things we don't have to process, things we don't have to think about per se. On the motor side, our efferent side, we then have, again, the conscious side of things being somatic motor. This is all of our voluntary skeletal muscle contractions. And then we have autonomic motor, which is all of the things that we don't have any control over. So this is smooth muscle, cardiac muscle, our glands, anything and everything that we need to be able to control that is not skeletal muscle. So the sensory side of things, we have our somatic sensory information. This is everything that essentially comes from just about outside of the body. And so this is things that we can touch, we can feel, information about the pressure that it pushes on our skin. Is it light touch? Is it deep touch? Is it vibration? Has the temperature changed? Where is our body? So proprioception is essentially where are all of our joints, our muscles, our skin in terms of space. Where do we know exactly where those things are? How can you touch your nose without actually having to see your nose? Things like that. And then we have the special senses, which is all of our information coming from vision, coming from smell, from taste, and from our hearing. And so all of that information is there as well. This is all information that we're processing voluntarily. This is information that we're conscious and aware of. The visceral sensory information, this is all primarily things that are inside of us. And so this is things that are inside the digestive tract, inside the blood vessels, inside the joints and the muscles and things like that. So we have unconscious proprioceptive information that is there as well for things. We do have some conscious visceral information that comes in. So we can feel things like when you drink something cold and it goes down into your stomach and you can kind of feel it kind of coating over the stomach itself, things like that. We can feel pain there, but that would be all along the somatic side of visceral information. On the motor side, we have our somatic motor once again. And so the somatic motor side of things is controlling only skeletal muscle. And this is all voluntary. So we are under control of those muscles. The autonomic motor is all involuntary. We have no control over that output at all. And so this is things like smooth muscle as it relates to our digestive tract, our blood vessels, the size of our pupils, anything and everything that has smooth muscle, cardiac muscle, and then the glands output. In order for sensory information to, in a sense, detect anything, we have to have any number of receptors. And so receptors themselves can come in a couple of different varieties. And so we have exteroreceptors. This is all basically information that comes from outside of the body, so external types of information that's there. We then also have interoreceptors, and those interoreceptors are giving us information that is internal to our bodies. And so for the most part, when we talk about things like exteroreceptors, we're thinking about the things that we can touch and feel and all of our special senses and everything like that. So once again, this is conscious information. Interoreceptors primarily are things that we don't really have a whole lot of control over for the most part. There are a few pieces of information that we can get from there as well. And then we have proprioceptors. Proprioceptors are both conscious and unconscious. This tells us everything about where exactly our muscles, our joints, our skin is at any given time. And so this is where when you have an itch on your skin, you can instantly kind of find it if you can also see it at the same time, especially. And you can get to it fairly easily. But you also know exactly where your arms and your legs are and your neck and your back and everything are at any given point in time. And that's all conscious information. But all of that same information from proprioceptive information also goes to an unconscious side of the central nervous system. The whole basis behind the nervous system is that we have neurons. And so neurons themselves have the ability to fire off action potentials. And so they're relatively excitable. It doesn't take a whole lot for them to fire themselves off. They do have a very high metabolic rate. They have a longevity that is essentially the length of our lifetime. And so for the most part, they are going to be with us from beginning all the way until end. Some of them will die along the way. And for the most part, they are going to be amitotic. They're not going to be able to divide, so we can't replace them. When we look at a neuron, the big part of the neuron that we can see is the cell body. This is also referred to as the soma sometimes. So the cell body is essentially the metabolic center of the cell itself. This is where all of the metabolic production is going to happen, especially in the form of Nissl bodies, where we have the production of neurotransmitters. And so this is where the substances that are going to go down through the axon and make its way all the way to the end of the axon, they're going to travel from the cell body all the way down to the axon terminals that are there. And so the cell body is going to receive information from other cells. That information is coming in along these dendrites. And if that stimulation is great enough, it's going to generate an action potential here in the axon hillock. So we have two major processes that come off of cells. We have the dendrites, which in this case we have a multipolar neuron. So we have many, many, many dendrites. And then we have a single axon. And so we have just a one axon and many of the dendrites that are there. Dendrites tend to be receptive, so they're receiving information. So information is coming into the cell from these dendrites. Axons, on the other hand, are outgoing. This information is traveling away from the cell body. And so this is where we're going to have our action potential, this electrical discharge occurring and flowing away from the cell. Sometimes axons do branch, rarely, but they can branch. When they do so, they branch in between these little nodes here, the nodes of Ranvier. And so that's going to get that communication to head out through the cell in that direction as well. So we just said that that particular cell type was a multipolar neuron. And so we had something like this where we had many, many dendrites and a single axon that came out of it. We can also have bipolar, where we have a single dendrite and a single axon that leave the cell. And then we can have unipolar neurons. They have a single process that comes out of the cell. It splits, and basically it looks like an axon no matter which direction we go. The only place where we can tell the difference is at the very ends, where it is either a dendrite or a axon terminals at either end. So we talked about that we had motor neurons, and we talked a little bit about sensory neurons, and bridging the gap between the two, we have interneurons. And so interneurons are the cells that basically kind of do the processing. This is the information that's going to get processed within the central nervous system. They can be as simple as just a interneuron that goes in between a sensory neuron and a motor neuron in a reflex, or they can be all of the different interneurons that we have within the central nervous system of the brain itself. And so this is going to essentially process the information as it comes in, and we determine what we need to do with that information from there. In addition to the neurons, we also have neuroglial cells. And so these glial cells, meaning glue, are the cells that kind of hold all the neurons in place, so to speak. They don't make any kind of action potentials, so they're not transmitting any kind of information, but they're basically just helping out. And so they're doing everything from grabbing onto blood vessels to grabbing onto the neurons themselves and holding them in place, to wrapping up and insulating those neurons, to wandering around and getting rid of debris or even infection that may be in the areas. So we've got a lot of different cells there. And in terms of the total numbers of cells, those glial cells far outnumber all of the neurons that we have. So we have things like astrocytes. Astrocytes come in and grab onto both capillaries and the neurons. They're going to kind of direct their growth there. When they grab onto blood vessels, they create our blood-brain barrier within the central nervous system, helping out in a sense with that blood-brain barrier and creating our cerebral spinal fluid. We have our ependymal cells, and so these are ciliated cells. They've got lots of little cilia that are on the top of them, and they're helping to move the fluid along within the central nervous system that's there. And that fluid is going to be the fluid that actually serves to give nutrients to the nervous system. We have neurolemocytes, and so these are our Schwann cells. And so these cover our peripheral nervous system nerves and insulate them. They are similar to the oligodendrocyte in that the oligodendrocyte creates this insulation as well, except for that insulation, the myelin sheaths is created inside the central nervous system. And then we have microglial cells. These are those wandering around macrophages looking for things that shouldn't be there within the cells themselves. And then we have satellite cells kind of maintaining the environment around the neurons themselves that are there. So our astrocytes here create the blood-brain barrier. And so that's one of their main jobs and their claim to fame in a sense that is inside the cells. They make up a large percentage of all of the central nervous system itself. They are just kind of like everywhere within the central nervous system. And so they're regulating what's able to actually make it out of the blood vessels so that we're not getting any kind of toxins or anything coming out of the blood vessels and touching nervous tissue. Microglial cells are phagocytes, so they undergo phagocytosis within them. Depending upon if we have brain injury, depending upon whether we have infection, those numbers are going to go up when we have either one of them. Ependymal cells, these are again ciliated cells, and so we find them within special areas in the brain called the ventricles, and then they're inside the spinal cord in the area of the central canal. And so these are ciliated cells that are going to help to move around that fluid that's inside there. Our satellite cells are kind of like regulatory cells, and so these are kind of monitoring what's in and around the cells themselves and are working to make sure that the neurons can function at their optimum. So one of the important things that happens within neurons is myelination. And so the myelination is the coverings that we have over our cells, whether they are in the peripheral nervous system or the central nervous system. So oligodendrocytes serve the central nervous system, and our neurolemocytes, or our Schwann cells, serve the peripheral nervous system. Essentially, those cells wrap themselves around the axons, and as they wrap themselves around the axons, they kind of squeeze out all of the cytoplasm and just leave the plasma membrane to cover over all of the neuron itself. And so we have this phospholipid bilayer that is many, many, many layers thick that helps to insulate the cell. And so for like a Schwann cell here, a neurolemocyte, the cell, it kind of starts out looking like a hot dog in a bun, and then all of a sudden the bun starts to grow. And as it does so, it wraps itself further around and around and around until it develops this big myelin sheath here. And so that myelin sheath can be very thick in some cells. It can be relatively thin in other cells. We have a little bit of cytoplasm and the nucleus and everything that stays on the outside of that Schwann cell that's there, and that helps to keep that Schwann cell alive. That's all the nutrients that it needs to maintain the cell. But it's this phospholipid bilayer thickness here that is what's going to increase action potential speed. This is going to insulate the cells, just like we have insulation on wires. If you take all of that insulation off, it's going to potentially short circuit, and we don't want that. And that same thing can happen with our neurons as well. A large percentage of our axons are myelinated, so they have that covering over them. And essentially what it does is it makes a faster nerve impulse, and so it's going to speed up the process. But the axon is not completely covered, so we have our little Schwann cells, or neurolemocytes here, and then running through them, we then have our axon, and that runs in between them. Each of the spaces that is there in between is the node. And so these are our neurofibril nodes, or our node of Ranvier, and this is what is in between the individual cells themselves. And this is the only place, those little nodes, is the only place where depolarization has to occur. And so our action potential can basically jump from one node to the next node and skip the majority of our neuron. Versus an unmyelinated axon, here we don't have that myelin sheath that's there. And so here, if we were to look at the end of a cross section of the neuron and the neurolemocyte, we're going to have something that has something like this in it, and then we're going to have multiple neurons attached to that neurolemocyte, but they're not completely covered. And so if we look at the side view, we have our neuron here, and then we have the neurolemocyte, and all of the axon is exposed, which means that it has to depolarize the entire thing. It has to go along the entire length, which is going to make it slower. And so we're going to have continuous conduction for our unmyelinated cells, and we're going to have saltatory conduction for the myelinated fibers. So here we have an example of unmyelinated fibers. So they are exposed the entire length of the axon. So that whole axon is going to have to depolarize as it moves along. In the peripheral nervous system, we have the potential for repair of damaged axons. And so those axons themselves, if they become damaged in any way, they become cut, they become torn, they become crushed, they become stretched, things like that, they have the potential that as long as the cell body hasn't been damaged, that axon may be able to repair itself and grow back to where it needed to go. And so it's going to go through a process where essentially everything distal to the damage, so this is the damaged portion right here, everything distal to it's going to die off. And so the axon itself is going to die off from that point. It's going to seal itself off, and the remaining neurolemocytes, the Schwann cells, are going to grow and create what's called a regeneration tube. This regeneration tube is going to essentially create a pathway for that neuron to get back to where it needed to be. Most of the time, many of the times, this can occur depending upon how far that process is. And as long as, essentially, if we think about a nerve, we don't just have a single neuron, but in a nerve, we're going to have many neurons. And so all of those neurons that were damaged at that point in the nerve need to, in a sense, line back up exactly with the other side. And so they need to get back to the exact same spot on there. Otherwise, we may have, if it's going to a muscle, we may have muscle weakness. If it's going to the skin, to sensation, we may continue to have a lack of sensation in that part of the body. So just like everything else we've seen so far, our bones and our muscles, we also have connective tissue that surrounds our neurons. And so we're going to have the whole nerve itself surrounded by an epineurium. And so this is going to surround the entire thing. We're going to take bundles of neurons and divide them up into fascicles like we did in muscles. And so that's going to be surrounded by a perineurium. So that surrounds the outside of an individual fascicle. And then we have surrounding individual neurons, we then have an endoneurium. And so that's going to surround the individual cells themselves and protect them as well. Especially in larger neurons, we're also going to have, or in larger nerves, we're going to have blood vessels. So we're going to have our blood supply to those cells as well. In order for communication to occur, we need to have synapses. And so when axons come to the next cell in line, this could be another neuron, this could be some sort of muscle, or it could be a gland. At that point, we're going to have what we call a synapse. And so this is the connection between the axon and that next cell that's there. When we look at it, we have a presynaptic neuron that is there. This is the one where the information, the action potential is heading towards the synapse. And then we have a postsynaptic cell or a postsynaptic neuron. And so that information there is then going to travel away from the synapse. In between the two, we have our synapse. We have the space that's in between the two cells themselves. And this is where typically neurotransmitter is going to be released from the presynaptic neuron onto the postsynaptic neuron and allow for that signal to travel to the next cell. So here we have our presynaptic neuron, and then our postsynaptic neuron. The space in between the connections between the two cells is our synapse itself. And so that lies in between the two cells. And inside that synapse, this is where our neurotransmitter are going to be released from the presynaptic neuron, bind to the postsynaptic cell, and then that's potentially going to cause an action potential in that postsynaptic cell. Synapses can occur in many different places. The most common that we think about are axodendritic synapses, but we can also have axosomatic onto the body. We can also have two axons communicate with one another, so we can have axonic as well. In addition to having chemical synapses where we have neurotransmitters going between the two, we can also have electrical synapses. And so electrical synapses are more common in the brain, but we don't really use them a whole lot, but they are somewhat there for those. This allows us to communicate one cell to the next by simply moving ions from one cell to the next cell. And so those ions move from one cell to the next cell, and we have then the inside of the cell going from negative to being positive as those ions move in, and then this postsynaptic cell starts to fire off an electrical impulse as well. And so this is more like kind of twisting two wires together. The two wires directly communicate with one another as they send their signal from one cell to the next. Chemical synapses, this is what we typically use both in the brain and the peripheral nervous system, utilize neurotransmitters, chemicals, that are going to gap the junction between the neuron and the postsynaptic cell, whether it be another neuron, whether it be a muscle, whether it be a gland. And so those neurotransmitters, in this case it's acetylcholine, what we used in muscle for getting the muscle to start to contract, that neurotransmitter binds to a receptor, and that receptor then has some effect on the cell. It opens up an ion channel and starts something going on in the cell metabolically, whatever it happens to be that particular neurotransmitter and the receptor cause to occur in that postsynaptic cell. That's there. The neurotransmitters themselves are just simply chemicals, and so in order to get that release of the neurotransmitter out onto the cells themselves, we have to have that induced, in a sense, where it actually causes the cells to release that neurotransmitter. And so as the electrical impulse travels along the axon, as it gets towards the end of the axon, calcium is going to start to move into the inside of the cell. So we're going to have voltage gated calcium channels opening up and causing the release of neurotransmitter into the synaptic cleft. That neurotransmitter then goes across the synapse, binds to receptors on the postsynaptic cell, and then that receptor that's there may cause the influx of sodium, which then takes the voltage from a relative negative charge and starts to bring it up towards a positive, and that then may start an action potential in that postsynaptic cell. This process of getting the neurotransmitter across the synapse and bind to the postsynaptic cell is what leads to what we call synaptic delay. Essentially, it takes time. And so with chemical synapses, we end up having a process where there's going to be a period of time of kind of delay of a few milliseconds, potentially there. Typically, it's hundredths of a millisecond that it's going to take, but it's still a little bit of time that it takes to get them to cross that synapse. The action potential as it travels along the axon, its speed is going to be dictated by the axon's diameter and myelin. And so the bigger the axon is, and the more myelin that cell has, the thicker the myelin that is there, the faster the transmission of the action potential is going to be. A smaller neuron with very little myelin is going to be a lot slower. And so it's not going to be able to transmit its action potential as far. If we take that small neuron, even if we have a large neuron, but don't give it any myelin whatsoever, this small neuron with myelin may actually travel faster than that larger neuron with no myelin. When neurons travel and kind of send their information, they send their information in oftentimes what we call pools. And so we can have different kinds of pools that occur. And so we can have converging pools where the information essentially is coming from multiple sources, and we're going to have one output. So we've got a lot of information coming in, and we decide on a single output. We can have diverging information where the information spreads out from a single input, and it goes to many different places within the system itself. And so diverging could be like hearing a sound or smelling an odor or a taste or something like that, and it reminds you of a whole bunch of things of information there. Converging information may be all of the different sensations as you're feeling something, and it's giving you a mental image of what you're touching. That's there. Reverberating feeds back in on itself, and so the original stimulus gets fed back in to itself and continues to stimulate it. It's kind of like a positive feedback loop. And then we have parallel after discharge. And so here we have inputs coming in. They're running along the same pathways, and then we have a similar output as it comes along. And so each one of them has a different function within the central nervous system of kind of communicating to many different neurons about the information that has been received.